running, recording on this computer. So, what you guys can see in front of you, again, remember we finished off today talking about kind of the specificity of neural applications, right? That things that are going to change, you're going to get better at doing precisely what that movement is that you've been training. Right? So, if you train with one arm, you get better doing one arm. If you train with two arms, you get better at doing two arms, et cetera, et cetera. When you train eccentrically, you get better, or you get more adaptations during eccentric contraction. So this one kind of reinforces the thing that we had seen previously. I want to draw your attention to what we see here. And what this is going to be, okay, is it's basically trying to show you here's the EMG signal, okay? And what you can see is that we're getting this kind of these changes that we're doing concentric contractions and eccentric contractions, that what we seem to be getting out of everything is getting these sort of larger changes, okay? In post-training, we're getting these larger changes than eccentric actions. So there seems to be, this is going to be a recurring thing, which is why I mentioned it here. There seems to be something magic, okay, about eccentric contractions. When you do eccentric contractions as a part of a resistance training regime, okay, whether that is eccentric only or concentric eccentric, so a kind of typical dynamic training action, you get better. That's probably a controversial thing, but I think you get better adaptations when there is eccentric somewhere that is involved, okay, in comparison to doing concentric only, which is fine because nobody really trains concentric only either. It's almost impossible to do all of that. But one of the most profound things that happen when you do resistance training that has an eccentric component is that the amount of muscle that you recruit, okay, during subsequent eccentric contractions as you become more and more trained increases dramatically, okay? And so that is going to have a very powerful effect on limiting muscle damage, exercise-induced muscle damage as we go forward. We really want to kind of briefly mention in this class. That's one of the big powerful things, which is why you go and you do squats and you haven't done squats in forever and you're sore, and then you come back like 3 weeks later and you do that same squat routine again and you're not sore again the next time because you have ramped up the amount of muscle activity during your eccentric contractions. You're using the same force, you're applying that same force over many, many more muscle fibers, therefore the actual mechanical stress. But as we will see, there's some things about causing hypertrophy and stuff that there's just something magic about eccentric contractions. This is going to harken a little bit to the data from Biopsy that I showed you guys. And again, this is just about specificity of training adaptation. Okay? I'm just trying to beat all of this stuff home to you guys that however you train, you get better at doing that. Okay? And so these are data on rate of force development. So what they're looking at is basically generate as much force as you can, as fast as you possibly can. And look at how we've condensed down our time scale here. This is what is, in essence, about half a second in time. Okay? So this is the front portion, basically, of starting a muscle contraction. And so here is what we're going to say, contractile rate of force development. So it's basically the slope of this line on the front side. Okay? And what you will see then is that both at early on, so 30 milliseconds, 50, 100, 200 milliseconds, and, okay, at peak, which is going to be the fastest rate that we're getting here, at all of these points after training, I can develop force faster. And that development of force, okay, is associated with an increase in the neural activation during that time frame and an increase in the speed of turning on of that neural activation. So the rate of the EMG change, the rate of force change is going up. And the total amount of EMG is also going up. Okay? So when you do resistance training, you get better at turning on your muscles fast, okay? Which has very, very, very profound impacts on sport performance where we need to try to maximize power and rate of force. Okay? Part of this is that you learn how to recruit those 2X motor units that are sort of not that often used in people that are sedentary. As you do that, then I get more force and I get that force faster. Or if I get more force in the same absolute amount of time, then my rate of development is going up. The math of that has to work. Okay? Now, this is not, and there's data on this, okay? This is kind of traditional training, 4 to 5 sets of 3 to 12 reps, okay? This is not training specifically to try to go very, very fast, okay? This is just sort of normal. It is, yeah, move the weight during these sets as fast as you can, but this is not lower, kind of moderate to low weight, like plyometric, really, really trying to go fast. This is just showing that even doing some of this more heavier traditional resistance training increases rates of force. Okay? That's very likely all a neural adaptation. There's no changes in, right, DNA. So again, we get these very powerful neural adaptations in some of these changes. Okay? All right, so to summarize, we are going to see increases in motor unit recruitment and muscle activation at max. So your 1 rep max, your maximal isometric contraction, your maximal eccentric contraction, more motor unit recruitment, more total EMG signal during these things. Okay? All of us, with maybe Keldon, who just works out all the time, have some untapped motor units in a lot of our things. Now, that's probably a gross generalization. Even Keldon, even, I don't know, Megan and Callie and everybody who does lots of things, Ryan, I know, who does a lot of resistance training, we all probably have some motor units that we're not used to. We're not very good at. So we can increase them. For a sub-maximal load, we see decreased activation of muscles. Okay? And that's going to be very reasonable. It's going to cost us less energy because we're just more efficient. Adaptations are specific to the type of training, concentric, eccentric, isometric, and at testing velocity. Okay? So when you train fast, you get necessarily better at going fast. When you train slow, you get a lot better at going slow, not as well as going fast. So if you want to go fast, you're going to train fast. Good point. Okay? We also see decreased antagonist activation, and we're going to see increase. Some of this all is driven by increased neural drive from the motor cortex and increases in excitability of motor neurons in the spinal cord. So more of whatever is coming out of the motor cortex is going to make it through the spinal cord. Okay? These are the things that tend to change in the first month, 6 weeks, 8 weeks of doing resistance training. And these may be the only things left to change once you've reached your genetic potential for increasing muscle size. Okay? You've reached your potential for increasing muscle size, which theoretically all of us have. Okay, now let's switch gears, okay? Now let's talk about the really fun rowish stuff, which is how do we make ourselves just jacked Okay, I want huge, giant muscles. Okay, how do I do it? Well, we call this a contractile protein adaptation. Okay, we are physically making more actin and myosin in our muscle fibers, in their nuclei. And we're going to integrate that additional actin and myosin into the sarcomeres. We're going to make more sarcomeres. Okay? We don't get more fibers, but our fibers get more sarcomeres. They get wider, they get fatter, they increase their cross-sectional area. That, in theory, should make them strong. Okay? So, the time course of all of this, it probably takes 6 to 8 weeks to see a measurable increase in cross-sectional area. Okay? It certainly takes 6 to 8 weeks to see something where, like, I'm going to look at Pearson and be like, damn, Pearson's getting huge. Okay? It's going to take some time for that to happen, which can be a frustrating thing when you're trying to get people to adhere to exercise. Okay? There's some evidence from, there was a previous study that they used Dr. Deb's PQCT, Dr. Beck's lab did, where they saw like changes in cross-sectional area. I think it was in the bicep, it may have been in the quad, in like 3 weeks or something. But there was an issue of they tested people pretty close when they finished doing their training session, and maybe it was due to swelling or something kind of after the bout of exercise. Some of the MRI data and stuff suggests it takes a little bit longer. So that's where we are. Everybody wants to know, how big can you get? Okay? We assume there is a genetic limit on cross-sectional area, but there's actually very little experimental evidence that either supports or refutes this, right? We assume one exists because there are just not people walking around, right? My dudes over in the Huff have been doing the same like curls and shoulder presses for 4 years since they've been here as freshmen, and they are not like, they don't all look like the Mountain or something on Game of Thrones. They're just not all these giant people, so we are going to make some assumptions. There's lots of animal data that suggests that. They train animals to do resistance exercise by like, they press on a lever that has some weight against it and it gives them food, right? And that little mouse will just be like, wait, give me the damn food. And they get to it and they stop. So most people see somewhere between maybe 5 and 40% increase in the cross-sectional area. Averages out somewhere between like 15 and 20%, kind of the middle point of our normal distribution. Okay? There is some genetics. We know they're going to determine that. There are some individual SNPs that we think are associated with all of that. And basically, like I could take a cheek swab from all of you, and we could see kind of what alleles you possess on certain things, and how you give you some evidence on. There's more of a chance that you're going to be a higher responder to resistance training than somebody that has different subsets. That's gained a lot of popularity. And like, let's take a cheek swab from my daughter and like figure out like, is she genetically going to be in a better place to be like an explosive athlete and something that benefits from resistance training, or might she be more beneficial for doing some kind of aerobic specific activity, right? Compares what she wants to do or what she actually demonstrates interest in, those things in. But you can pay, like people, you can take a cheek swab, pay folks a couple $100, and they'll get you this data out of all of that. If you do, I don't know if it's Ancestry, but one of the other ones, maybe it's 23andMe or something, they sell your DNA samples to drug companies. It's in the fine print. And they can tell you, like, are you at increased risk of type 2 diabetes, increased risk of cardiovascular disease, based upon all that, which is very, very useful information. They may also tell you things like that. You have to do those for these kinds of things. So we have some understanding of these things. If you care about that, I'm happy to give you some papers and kind of lead you down that particular thing. But the question is, who cares, right? Like, does it really matter? Like, if you're a responder, most of you would probably know if you're a responder by now, right? Because you've done some sort of exercise. You either are like, I got swole, or I did a whole bunch, I didn't really get very swole. So we kind of have an idea. We do know that when we give you steroids that all bets are off, and that's one of the other issues that kind of we have to deal with the upper limit, is Lots of big people that have the largest muscles that we've ever seen, humans, are very likely to take steroids. So we don't fully understand, and it's not, at least at this point in our lives, at this point with IRBs, it is not ethical for us to give people steroids, right? Because we know that there are the potential for some fairly significant harmful side effects to all of this. And so it's hard for us to do kind of randomized or real research studies to try to see what it's about, right? There are a couple of studies on steroids where they've given it to people. One of them is published in the New England Journal of Medicine. I'd be happy to give that to anybody who's fair. And it's just what you think. If we give you steroids, you get giant. If you get steroids, you need to do anything. We give you steroids and you work out, you get really fit. So, in that way, I mean, we're going to talk about hormones and stuff later. Probably maybe not today. It may be next Tuesday. We're going to talk about that there's some, and I will tell everybody now, be very careful when you talk about the dev about whether there's the effect of acute changes in hormones after bioresistance exercise, and whether or not that does in fact play a role in the training response and hypertrophy response that you see. She is more of the old school opinion, and there's plenty of evidence to support this, that yes, they do play some role. There's a newer line of research out of Stuart Phillips' lab and stuff, Master, says that the acute change after bout of exercise doesn't matter that much. As long as people have steroids, like you have your own, you make your own testosterone and estrogen things, that you're going to get whatever your response is. It's a kind of external, like a hyper physiological dose is what really changes things. The fact that Jacob gets a bigger testosterone response after bout of exercise than Alex does, and Golden gets like an even bigger response than Jacob, doesn't mean necessarily that that's going to be why Golden has bigger response. So there's some interesting stuff there. So here's this from that same study of almost 600 people. I showed you guys they were doing bicep training. We've got men and women. Here's this kind of rough normal distribution, the percent change across sectional areas. So most people kind of fall right here in the middle between maybe 10 and 25%. Okay? But there are some people that are out here on this long tail that are getting really, really big. There are some people that really don't show a lot of response to everything. There's a host of reasons why that could be the case. Okay? So what we need to understand, though, is what are the things that control all this? Why is one of the responses to resistance training? Why is one of the responses to loading a muscle, stretching a muscle, applying these things to it? Why is the response to make that muscle get bigger? Okay? And what are going to be the things that contribute to all of that? How do we turn that signal on? What are the kind of signaling steps? And what's the benefit of it? That's what we're going to try to kind of get at from a contractile protein. Okay? So before we get into the midst of this, I think it's really, really important that we take a few moments and talk about the models that people use to study resistance training adaptations, the models that people use to study hypertrophy following resistance training, and the strengths and weaknesses or limitations of each of those models, because we see different things depending upon which model that we use. And there are issues of the generalized ability, so the ability to take, say, something from an animal model and say that that's going to happen in people, or take something from an isolated cell culture and say that's going to happen in an animal model, which is going to happen in people. Even if the cell culture is, I take a muscle biopsy from Jacob, and I'm going to culture some of his muscle fibers. I've got his fibers, and I may be able to make them do something in the cell culture that may or may not happen with these fibers. Right? So studying resistance training is incredibly difficult. It's incredibly, incredibly difficult. And so because of that, it's one of the most widely studied things, but there's so much variation. It's really, really difficult to replicate studies and things because everybody does it a little bit different. And it's hard to have really good type controls, especially in human studies. In addition to all that, in addition to the experimental model concerns, there's also going to be issues on how do we measure and quantify just how much bigger your muscle is. So there are what we call invasive ways of measuring muscle size, and there are non-invasive ways of measuring muscle size. I think this is a really important distinction because there is good and bad or good and limitations to all. Okay? So an invasive way means I am physically going to harvest either some or all of the muscle out of your arm or leg or out of some animal. Okay. We can take the whole muscle out. Okay. I don't imagine that Ryan is going to volunteer to let me cut out her entire bicep. And then we can measure what we call myofibrillar cross-sectional area or total protein content. We can take out her entire bicep, grind it up, okay, and then use immunohistochemistry to pull down all the actin and myosin from that ground up muscle. Very, very accurate, okay? But how do you do that before and after training? I can't take your muscle out and grind it up and see how much is in there, and then magically put it back in there and then train it and check it again. That creates problems, right? Maybe we could do her right arm versus her left arm. What we typically do this in is in animal models where I can have genetically identical animals that are going to be fed exactly, I have a lot more control. They're identical, they're getting the same food, they're doing exactly the same thing, or I can restrict them so that they can't do things, and then I can much more easily compare my groups. If I take a biopsy or a piece out of somebody's muscle, I can take a little pencil eraser-sized piece of muscle out of Callie's leg, and I can get under the microscope and I can look at the individual muscle fibers and the cross-sectional area of those individual muscle fibers. Fine. I've done that. I've looked at them under the microscope. You can measure them very accurately. The problem is, what if in Callie's muscle, I just happen to grab muscle fibers that either got a lot bigger than the ones around them, or I got some that didn't get bigger at all and all the other ones did. Okay? So when you take a biopsy, either from an animal or from a person, we are making assumptions that that small little tiny bit of muscle is indicative of or representative of what happened in all of the rest of that entire giant muscle. And that's not an unreasonable thing to assume. But it comes with some limitations. Okay? So we have to keep all of that. I can take the biopsy and grind the biopsy up. And I can look at what we call myofibrillar protein concentration. How much actin and myosin were in your biopsy? Well, I take a biopsy before Callie trains. I then have her train, and I go back and I take a biopsy from a very, very similarly located place in her muscle. But it's not the same muscle fiber. And I'm going to grind up and look at how much protein content is there. Again, it's not an unreasonable thing to do, but we don't have as much experimental control there as we want. Plus, is that biopsy still indicative of the whole muscle? Because the Lord knows I can't cut out the whole muscle. Okay? So, what are our solutions to this? Well, in people, we do in the animal models of things, we just kill the meat. We just take the whole muscle. Right? Part of why I'm not an animal researcher. Not that big of a leap from that rat or that mouse to my cat. It's not that big of a leap from that cat to my dog. And yeah, right? There are necessary things. We need to do animal research. Like, I appreciate all of that. And it's a place for us to check safety. We learn a bunch of things we have control. But it's just not for me. Right? It's just not for me. Jacob?
Just the way you described doing research on rats as the gateway animal.
Right? They're the gateway animal to. There are people that do research on dogs. of the best exercise physiologists that I know. He's the editor-in-chief of our ACSM's flagship journal. He's at Auburn University. Bruce collects data. Bruce Gladney does, like, he knows as much about lactate as anybody. Bruce collects data like 2 days a year to do these dog experiments, right? Everything they do is published in the Journal of Physiology, the Journal of College. Really, really good. But they do these experiments on dogs. They have to kill the dog. And I'm just like, nope, I'm out, right? I'm out. I will fail, okay? And it's good research. I appreciate what they do, how they do it. I can't do that. Yes, a mouse is a gateway animal into that. There are people that do research in private, right? The monkeys know what you're going to do to them. They learn. They throw their shit at you, and those kinds of things. So, again, some of this is necessary and all of that. I appreciate all of that. It's just not for me. Okay? So in people, what do? We do this non-invasive stuff. Okay? So I'm going to bring Ryan in, and we're going to put her on a table in the lab, and we're going to stick the ultrasound machine over her muscle. And we're going to measure the thickness of a spot in her muscle with the ultrasound. And we're only going to be able to measure that thickness as much as, as deep as the ultrasound can go. So I can get an idea of what's going on, like in her vastus lateralis, just in her vastus medialis, right? I can't really get much of what's happening on her vastus medius or any kind of non-superficial muscle. Again, very reasonable thing, but, well, I got it in one place. What if that one place is indicative of the length of the whole muscle? What if that one muscle, that superficial muscle, isn't indicative of all the other muscles that I can't measure? There's just some issues. I can solve some of that by putting her in the magnet and taking an MRI. Works really, really well, very super accurate, okay? I still have MRI images from when I was a PhD student that we need to analyze. We could publish a paper on it, we not analyze, right? Because it takes so damn long to do. Very, very accurate, but super labor intensive. Plus, you got to go spend $800 or $1,000 an hour to rent a magnet and put somebody in there. Seems a little, a little expensive. It's fine with the drug. When Procter & Gamble wanted to pay us to use the magnet, no problem. They have deep pockets. I do not. Okay? So what then if you do the magnet, you have an individual like cross-sectional area versus the volume of an entire muscle. There's a whole bunch of, whatever we do, there's problems with all of it. Okay? I just want you to appreciate that. I'm going to show you data, and I will try to tell you this is from an animal model, and this is the whole muscle. This is from people, and it's individual myofibular cross-sectional area. This is from people, and this is from ultrasound, or this is from MRI. We have, downstairs, we have ultrasound. We can measure ultrasound things. We've done that. In Dr. Deb's lab, there's also the peripheral CT machine, so we can measure some individual cross-sectional areas, especially in like the calves or the bicep or the forearm flexors or something. We can do a little bit of quad as long as your legs aren't super big. And those things. So we can get some data from all of that. It's good data. It's very accurate, but again, it just comes with something. Okay? So hopefully now that I've convinced you guys that it's impossible to study all of this, now let's see some real data. Okay? All right. Now, to understand hypertrophy in some ways is kind of like VO2 max. Okay? This is an integrated process. There are going to be multiple cellular and molecular signals, all of which are going to work together to contribute to hypertrophy. Okay? So what we need to do is place some sort of stress on the muscle, specifically by performing resistance training, so that we activate the cellular and molecular pathways, so we activate the right ones in the right sequence, okay, that will then tell the nucleus of the cell to make more protein, make more actin myosin, make us more sarcomeres, so our muscle is bigger. Okay? Now, one of the things that we think are involved, okay, as part of a resistance training program that are going to be the stimulus or the signal that will activate these cellular and molecular pathways? Well, load. We think load is important, okay? We think the peak amount of muscular force or torque that is generated during a bout of resistance training plays some role in it. There's some evidence for that. We think that the total volume of your training session may play some role. So volume in this instance would be the amount of force you generate during each rep and the number of reps that you perform, okay? So some people call this volume load. There's some evidence that if you make volume load go up, it will do this. That gets to be a little tricky. There's some evidence that the contraction type matters. Okay? If you're doing concentric only versus eccentric only versus isometric only versus a dynamic thing that has mostly where you're going to lift it up and lower it down, that there may be some differences among those things. Right? There's evidence that the metabolic demand is placed upon the muscles during resistance. There's evidence that going to failure, reaching high levels of lactate, reaching a place where you are invoking peripheral fatigue, okay, that the metabolic demand may matter. There's different ways that we can manipulate the metabolic demand. I'll show you guys some interesting data on all that. This way. The local, so in the muscle, you might call things, we might call this like the autocrine or paracrine response, as well as the endocrine response, the systemic, whole body, hormonal, what I'm calling milieu, okay, which I just think is a great word. But all this means is there may be some evidence, and there's some that's there may be some evidence that release of hormones by the muscles and then release of other hormones systemically, if all of those things are around and available for the muscles, that they may also be necessary or at least sufficient to activate these cellular and molecular pathways. So because of all of this, it's complicated as shit. Okay? And so the problem that we run into when you have an integrated process like this, okay, is that most studies try to isolate the independent contribution of only one of these factors. They're going to try to hold load or peak force the same, but then manipulate volume while holding contraction type and metabolic demand and everything else the same. Or we're going to manipulate total volume, but nothing else. So we're going to have all of this the same, but we're going to manipulate contraction type. Okay? And people question. This is called a reductionist model of doing research. It's a very good way to do research, right? Everything is the same, manipulate one variable, see what its independent contribution is. But there are people that will argue, and I think they make a reasonable point, right? That is that an ecologically valid method to study hypertrophy? When I say ecologically valid, I mean, does manipulating only one of these things truly represent how people train in the real world, how athletes train in the real world, right? And that, well, I'm going to change one thing, but I'm also probably going to change something else. And so I can isolate this one thing and see this, but nobody actually trains that. I can tell you guys, there is some evidence that doing eccentric only training may, in a lot of ways, be particularly beneficial for hypertrophy. But most people don't train that way. You have to have a part, like it's incredibly difficult and labor intensive to train that way, and you can't do it all the time, or you will just like kill yourself, right? You'll destroy your muscles. So, you know, just because I can do it in the lab and show something doesn't mean that that's actually of any use to a person who does this in practice. So just keep all of that in mind as well. Okay? All right. So to get more protein, to get bigger muscles, I must tell the nucleus of my cell, okay, to begin making more protein. That begins with this process called transcription, okay? I take a DNA molecule, I get some signals into the nucleus, the DNA molecule opens up, and I begin making messenger RNA. This is this process of transcription. So I transcribe messenger RNA. Those messenger RNA are then going to get out of the nucleus, out into my cell. They are going to bind onto a ribosome, and then that ribosome is going to bring in amino acids, and based upon the code on the messenger RNA, it is going to put the amino acids together in a sequence that is then going to translate the RNA into a particular protein. Okay? Now, I assume that most of you have had this idea of how we open this up, right? We're going to have the different enzymes, which I cannot remember right now, and all my old biology teachers are rolling their eyes at me. DNA, something, translocase, or I don't remember. Okay? But this process, if you want to know more about it, it's very, very involved. Is less important for what we're going to do in our class. And I just need you to appreciate it. We need signals to tell the nucleus to make the messenger RNA. And then we need the ribosomes to turn the messenger RNA into the protein. Okay? One of the ways that we learn about what's happening inside the cells is we do a reaction called the polymerase chain reaction. This is what you do when you're going to go get tested for COVID, and they jam the thing up your nose, they swab it around. What they're looking for there is they are looking for messenger RNA that are going to code for a particular protein. And they take that messenger RNA, and in the polymerase chain reaction, they essentially make protein out of the messenger RNA from a tissue. They can measure the RNA content has gone up, which is going to tell us that the nucleus has begun making more and more RNA, and then we can use those individual messenger RNA and see what kinds of proteins we make from all. So if messenger RNA are going up, we make the assumption, so if you've got messenger RNA for myosin, they're going up, we assume then the cell is going to make more myosin, right? We've not actually measured that there's more myosin, but our assumption is that there's going to be more myosin. So a lot of isolated cell culture things, what we're looking for basically is the nucleus gets activated, these RNA levels are going to go up. Sometimes they measure for the actual protein, sometimes they just measure for the messenger RNA. It's the same kind of thing. I'm measuring messenger RNA for testosterone coming out of the muscle. Right? So that's kind of what's going to happen. Okay. We'll probably finish talking about protein balance, and then I'll turn you guys loose because if we get into the actual data, then there's not a really clean stopping. Okay? So if this is how we make protein, we need to appreciate this idea of what's called protein balance. Okay? My father, the ex-football coach, one of his favorite quotes, maybe I've told you guys this, I forget, I only have all 5 stories. Someday that will be you guys. You'll be teaching and be like, wait, did I tell that story? And your students are like, yeah, she fucking told us that story. Stop telling us the same story. One of his favorite stories was this relatively famous football coach, it was Lou Holtz. Coached in Arkansas for a while. He coached the New York Jets. He coached Notre Dame. He was out throwing these things. He was on TV kind of as a caricature of himself. But Lou used to say, right, that you're either going to get better today or you're going to get worse today. Never say the same. Right? So I'll ask all of you guys, as master students, did you get better today? Did you do something productive? Did you learn something today? Or did you get worse today? Right? As a faculty member, I probably got worse today. I just chased my daughter around. I haven't done a lot. Well, Ryan and I had what I thought was a relatively nice conversation about what we're going to do for her thesis. Maybe I did something that was productive. Okay? Protein balance, your muscles operate in a similar manner. Your muscles are either getting bigger today or they're getting smaller today. Okay? I hate to tell all of you that. They're either getting bigger or they're getting smaller. And so this is this idea of protein balance. There's always this constant state of flux where we're either driving synthesis of muscle-specific proteins, okay, based on what you've eaten, based upon if you took steroids, based upon if you've done some sort of resistance training or activity, or let's say you haven't eaten anything all day, you're in a caloric deficit, right? So you're going to drive protein breakdown. Hormones are going to drive the breakdown. Cortisol is going to say, hey, let's start stripping out amino acids out of muscle so we can send that to the liver and make some glucose. Because the brain needs some glucose. Who gives a shit about the muscles? Okay? So levels are going up and down every day, and it may change. Usually going down in the morning, right? If you haven't eaten for a while, anything. So when you're in a positive protein balance, so synthesis predominates, your muscles get bigger. That is called hypertrophy. When you're in a state where breakdown is going to predominate, that's a negative to protein balance, then your muscles are going to atrophy. Okay? And so if we want to experience hypertrophy, this is why it takes so long to see it, is we need to be in a positive protein balance in skeletal muscle for weeks, okay, so that we can accumulate enough of an increase for it to actually be noticeable in that muscle. So they're always in this state of flux. So what we need to do with exercise is do something to tip the scales towards hypertrophy in a way from atrophy. Okay? So an increase in protein synthesis is called anabolism, and an increase in protein degradation is called catabolism. So we need anabolism to predominate and drive hypertrophy, rather than catabolism, which is driving further breakdown. Now, you guys may have heard catabolism when it is sometimes called, which what happens with aging. It's sometimes called ataxia. It's sometimes called sarcopenia if it's an older person. But the process is the same. Basically, you're losing muscle because we're breaking out, we're stripping out those proteins to do something else. Okay? So we regulate protein balance. We regulate the balance of anabolism and catabolism through hormones, diet, what we call mechanical loading or resistance training, stretch, okay, which is going to be a weird one, metabolic stress, and potentially nervous system activity. I put a question mark here, but there is some data that also plays a role. Okay? So what we're going to hope to do if we want to make our muscles bigger is mechanically load them, ideally while getting some stretch during that load, and make sure that we have an adequate amount of protein in our diet, that we are, even though it may not matter a huge amount, that we at least have enough hormones going around. Our nervous system will be active correctly. This, we work metabolic stress, metabolic failure. We can push all of these things together to drive anabolism. But then we still have to do it over again. As soon as you stop, I'm talking within days of stopping, it immediately goes back the other direction. Okay? Immediately. There is space flight data from people going to space for 3 days. And there's like 5% loss of muscle because there's no mechanical loading. You lose it. If we want, we can actually measure protein synthesis rates before and after training, right? Whether you're at rest, before and after a certain kind of meal. But we can measure protein synthesis rates. But I will warn you guys that changes in the protein balance rate, changes in synthesis and degradation rates, sort of acutely, like after one bout of things or after one meal or something, do not match up very well with what we see with prolonged caloric deficits, prolonged resistance. Okay? And with that, I will call a stop to this. With that, I will say, have a safe and happy Thanksgiving. It's Thanksgiving. Go eat a bunch. But exercise tomorrow. Okay? If you exercise tomorrow, you'll have more sensitive GLUT4 receptors. You'll have a more active LPL, lipoprotein lipase enzyme, so you'll metabolize more fat, and you'll be able to stick more Resynthesis of glycogen, stick more of those carbs into your muscles rather than going to fat. So exercise tomorrow, eat like a champ on Thursday. Okay.